Hey guys, this is Mrs. Holmgren. Today we're going to be talking about viruses. We're going to talk about the structure of viruses, types of viruses, and the way that viruses reproduce lytic and lysogenic cycles. When we talk about viruses, the first thing that we need to note is how tiny viruses actually are. And if you take a look here, we've got centimeters, millimeters, micrometers, and then down here we end up with our nanometers. And so plant cells, animal cells, these are very small cells. They're measured on a micro scale. Bacterium are also measured on a micro scale. And then here are our viruses. Viruses are actually smaller than bacterial cells. And viruses are measured in nanometers. You can actually only see them with an electron microscope. So they are smaller than monarins, which is another word for bacterial cells. Viruses are unique in their shapes. Viruses can have a spiral shape like you see here, kind of like a Lassa fever would have a spiral shape. They can be polygons. You can see there's a polygon here that I just pointed to. With kind of spikes coming off of them. If you've ever played Scattergories, the dice that has all the letters on it, that would be a polygon kind of shape that a virus would be. There's rod-shaped. The tobacco mosaic virus is a rod-shaped. They have bullet shapes. Look at how weird this guy is right here. Some of them have a needle-like shape to them. And then one very, very unique type of viral shape is pictured right here. This is a bacteriophage. Bacteriophages are viral viruses that will infect bacterial cells. As I said at the beginning, we're going to talk about the parts of the virus. The first part here is the capsid. The capsid is pictured right here. Capsid is the outer protein coat. This is what allows the virus to enter a cell. So the capsid is going to actually bond to the membrane of the cell that it's going to infect and kind of trick the cell into thinking that the virus is something that the cell needs. The next thing, inside of this capsid, you're going to find the DNA or RNA. There has to be some type of nucleic acid in order for the virus to insert itself into the host cell's DNA and then make copies of itself. Finally, an optional layer is the envelope that you would find around the capsid. The envelope is actually a part of the host cell membrane. It just kind of allows the virus to be a little bit trickier and fool the host cell a little bit better. And then the host cell will take in that virus, and the virus will become a part of the cell's DNA. So the envelope is an extra layer. Usually these are going to be in animal cell viruses. Objective 10 says to distinguish between the types of viruses. So we have basically three groups of viruses. Bacteriophages, DNA viruses, and then RNA viruses are called retroviruses. And so we'll kind of see these two words used interchangeably. Here's a bacteriophage. This is a simple one. Bacteriophages are viruses that infect bacterial cells. So this is the virus, the bacteriophage. This here is the border of a bacterial cell. And over here, all of these tiny dots that you see, these are all bacteriophages inserting and injecting their DNA into the host bacterial cell. They kind of look like little spaceships. They have these legs here that they attach to the host cell. Inside of here we would find all kinds of DNA that it literally injects almost like a needle into the host cell. DNA viruses, pretty easy definition. They get their name from the fact that they have DNA as their genetic material. Some examples here are the canine parvovirus, which your dogs would get a vaccine against getting parvovirus. Another one is the chickenpox vaccine. DNA viruses are pretty stable. Their DNA doesn't change a whole lot, so DNA viruses are very easy to vaccinate against. More examples of DNA viruses. The herpes virus, this is the herpes virus right here inside of a host cell. The common cold virus, adenovirus, Again, DNA virus and human papillomavirus causes warts. This is a picture of the virus by itself. Again, most of these DNA viruses you can get a vaccine against because their DNA is stable and does not change. These RNA viruses are where things tend to get a little bit tricky. Examples of RNA viruses: the common cold, measles, mumps, all of these lines that you see inside of the cell, that's the virus that's taken over the host cell. The rabies virus here, again, these that you see on this slide, measles, mumps, the rabies virus, these are things you can get vaccinated against, but we don't get a vaccine for the common cold, and we'll talk about why that is in a couple of slides here. So the retroviruses are RNA viruses. They have RNA as their genetic material. Here's where it gets tricky. These viruses have RNA as their genetic material. Well, a cell does not know, the cell cannot integrate RNA into its nuclear DNA. So what has to happen here, in order for the retrovirus to become a part of the host cell's DNA, the RNA has to be converted back into DNA, and then it's used to make RNA again. So normally what you do is you have DNA that's converted into RNA, and then you make a protein. Okay? So that's how it works normally. But in the case of these retroviruses, the retrovirus has RNA. It has to integrate into the cell's DNA, so it has to get converted into DNA. Then it gets converted back into RNA, and then you'll actually make that protein that you wanted to make. So this step right here, this RNA to DNA, uses something called reverse transcriptase to convert the DNA into RNA. And when that happens, when it works, this is where mistakes can be made. And we end up with the virus mutating. And that is why diseases like HIV, diseases like the common cold, these diseases we cannot vaccinate against because the DNA, that conversion between the RNA to the DNA, mistakes get made and the virus mutates. And so it mutates faster than we can kind of keep up, with the vaccines. This is a hot topic in science, whether or not viruses should be considered living or non-living. The reason why a virus would be considered a living particle is because they do reproduce, they contain DNA, and they do evolve, especially the retroviruses. But when we look at this non-living, they have to have a host. And why do they need a host? They need a host in order to reproduce. So even though they do reproduce, they must have a host cell in order to do this. The other big thing here is that they do not have cells. Viruses are not made of cells. They require host cells in order to survive. This is a hot topic in science. Personally, I would say that a virus is not a cell because of this reason a virus is not a living organism because they are not made of cells. And that's one of the facts that we learn in science, is that all living organisms are made of cells. Just a couple more things about viruses. Almost all organisms have at least one virus that infects them. As you've seen, bacteria have viruses that infect them. Protists will have viruses that infect them. So the simplest organisms even have a virus that will affect them. And viruses are host-specific. So what that means is basically the capsid or the envelope, so that's the capsid, this green layer is the envelope, is specific for a host's receptor proteins on the surface of their cells. So if you have a cold or influenza, you're not going to give your cold to your dog because your dog has different proteins on the surface of their cells than you do. And like vice versa. If your cat seems to be sick, you're not going to catch a cold from your cat because your proteins on your cells are different than your cat or your dog proteins. Our last objective for the day is to distinguish between the reproductive cycles of viruses. They reproduce in two ways, lytic or lysogenic. So each virus is different in the mode that it reproduces. So we're going to go ahead and talk about the lytic cycle first. Your textbook describes a lytic infection as the outlaw. Simply stated, the virus is going to enter the cell, it's going to replicate and cause the cell to rupture. Why is the cell rupturing? The cell's rupturing because basically it's becoming full of viruses, and so there are so many viruses in the cell that it just simply ruptures. So what happens here with the lytic infection? One, the virus lands on the host cell. Two, it injects its DNA into the cell. So if you look down here, we have a bacteriophage injecting its DNA into the host cell. Three, the virus is going to basically take over the cell. Whatever the cell is supposed to do normally, it's going to shut it down and start making just viruses. And so those viruses just get assembled, and the cell becomes just this factory for making viruses. And then lastly, the cells become so full of viruses that it will rupture or burst. And the science word for that is to lyse, means to rupture. The other way that a virus can reproduce is through a lysogenic infection. This is a kind of strange one. The virus will actually insert its DNA into the host's DNA, and then nothing happens. So the viral DNA is just hanging out in your DNA, a part of your DNA. We call it a prophage. That's the viral DNA that's a part of your host cells. And it just waits. So every time one of your cells divides, the viral DNA will get copied with it. And the viral DNA just gets copied every time one cell makes another cell through mitosis. The viral DNA just gets copied. So you don't even know that you have this virus. You're not sick. It is not, it's not carrying out, it's not making you sick. Here's our viral DNA. Here's that prophase. The viral DNA is a part of your DNA. And then every time one of your cells divides, it's going to copy the viral DNA with it. So here's the original cell that becomes infected. And then, here's the prophage inserted into that cell. And this cell divides here. And then this cell divides again. And so now you have a multitude of cells that all have this viral DNA in it, kind of waiting for a trigger, waiting to go into this lytic cycle where the viral DNA takes over the cells and starts to copy itself. The most common lysogenic virus that you guys would be familiar with is the herpes virus. The herpes virus it causes cold sores or fever blisters on a person's lip or in their mouth. The herpes virus will kind of hang out in your nervous system and just kind of wait for you to be stressed or go out in the sun or have a fever. And so once that happens, once there's a trigger, then the virus will start making copies of itself and it'll kind of expose itself, as a fever blister in your mouth or on your lip. I just want to talk briefly about these other non-viral particles that can cause diseases. Viroids are just a strand of RNA. They usually cause plant diseases. There's no capsid, so that's why it's not a virus, and there's no cure. One example of these viroids is the spindle tuber viroid that causes the potatoes to have these kind of oblong shapes. What happens with this is that the potato inside becomes discolored, and so you can see here are those viroid particles that would infect the plant. The other non-viral particle that we talk about here is a prion. A prion is just a protein. The most kind of common thing that you would have heard of is mad cow disease. Again, this doesn't contain any DNA or RNA. It's simply just a protein that accumulates in the brain and causes mad cow disease. The human version of this disease is Creutzfeldt-Jakob disease. You can see this is the brain of someone who has Creutzfeldt-Jakob disease caused by that prion, and chronic wasting disease occurs in animals.